So I'm Laura Webb. I work for EPA Region 7 in Kansas City, and today we're going to talk about a project where we use next generation qPCR to monitor for toxic cyanobacteria. A brief outline, or talk a little bit of background about the project, and then go into some new techniques that we've been developing and working with here in the region. Our project team was a 2017-2018 project. We had several folks from here in Region 7. Our laboratory here, and then Dr. Jingrang Liu from ORD in Cincinnati, who the bulk of the work presented is his qPCR work. And then Dr. Heath Mash, also in Cincinnati, did the cyanobacteria toxin for us. Goals of the project really were to develop a good screening tool to determine if what genus or species of cyanobacteria present, and then to determine if they're capable of producing toxins. We used a commercial kit here in the region to do this work. And then, like I said, Dr. Liu used qPCR there in Cincinnati to do the qPCR work that I'm presenting here. And then we really wanted to correlate the molecular and biological results with chemical results that we had. These are just a brief shot of the watersheds that we were looking at. We chose 2 lakes that represented reference conditions, very small watersheds, and then 3 lakes were our target lakes. And the one we're going to look at today is Lake Perry there towards the bottom right of your screen. It's a medium-sized watershed there in the state of Kansas. Our sampling plan, we were there at the lake each week on Tuesday for 26 weeks for both years, from April to October. We did surface water sample collection, 3 locations at each beach, and then composited those samples and brought them back to the lab. We had a large suite of analytes we were looking for, and everything from metals and PAHs, pesticides, down to cyanotoxins, both by ELISA and LC-MS. We did biological work, looked at E. coli as well as cyanobacteria under a microscope, did the molecular work, and then in situ parameters. This is just a look at the Lake Perry watershed. And one thing I wanted to point out, that arrow there is pointing to the Kickapoo tribe. They're in Kansas, and they play a crucial role here in a minute in the presentation. So I just wanted to show you where they were on the Delaware River upstream of Lake Perry that we're going to look at. Okay, so our first tool that we're going to talk about is Cyan. It's a collaboration between USGS, EPA, NOAA, and NASA. And it's basically looking at satellite images and doing some algorithms on those to get a picture, color picture of what what blooms are on the lake. And we're looking for a specific signal for cyanobacteria. So here's just a brief shot of some of the satellite images during the 2017 season there on Lake Perry. And you can see the more intense the color is, the more towards red it is, the higher the cyanobacteria concentration in the lake. If you look on the very left of the screen, you'll see I'm pointing to our sampling site on the lake. And you can see there are times during this growing season that we had a pretty good signal on the cyan data to show that we had a bloom in place where we were sampling. So this is kind of a composite of all of these tools that Cyan has available to us. One thing to note is, like I said, the Kickapoo tribe upstream on the Delaware River reported that they had a bloom just where their drinking water intake was, and they called and told me that in early August. So that river then flowed into the lake, and you can see that bloom develop in the northern portion of the lake, and then move down into where we were sampling, where the star is there in the middle picture. So we had an early warning upstream that bloom was going to develop in the lake. And sure enough, the cyan imagery then showed that bloom had showed up. The picture on the right there is the actual cell phone app that they developed, Cyan, the project developed. And you can see that it had a spike in the cyanobacteria concentration there in mid to late August. And that was confirmed by the picture on the bottom there. That is the ELISA concentrations of microcystin. So the bloom not only developed on the satellites, but we could also see that toxins developed in the lake and they all correlated to the same time. So it's kind of a really good confirmation that all these techniques were working together. Okay, so this is the same graph of the microcystin concentrations by ELISA on the top, and what you see below are the two pigments that we measured. The green is the typical chlorophyll pigment, and it did show higher concentrations during the summer months, which you would expect as the growth of the lake. And then the blue line there is phycocyanin that we measured when we collected our samples. So you can see we got a phycocyanin spike just prior to detection of the toxins in the lake. So what this really showed me was you can use chlorophyll, and kind of it's a gross explanation of what's happening in the lake, but the phycocyanin signal really popped for us right before we saw the bloom in the lake and the toxins. So we really like using phycocyanin to kind of give us a heads up on what's happening in the lake. And those were just phycocyanin measurements from the grab samples, not an in situ sonde. Okay, so here again, you've got the ELISA graph there on the bottom. You see I marked where the phycocyanin detection was. And then we're going to look at the qPCR results there at the top. So this is going to show us the potential for toxin production. It's a gene sequence that shows us that there's a sequence there in the water, in that DNA, that we could have toxins produced. And Xingrang used two different indicators here. The dark blue line is the MCYE, pretty traditional, used to show microcystin toxin production. The light blue line is an MSR2 is what he called it, and that is a microcystin produced by microcystis cyanobacteria. And both of those lines clearly show, they kind of match each other. So it looks like microcystis was present and it was one of the toxin producers. And we see a signal there on the qPCR results prior to seeing the toxins in the water. And so that's what we wanted. We wanted to be able to see a detection and get an earlier warning of when we potentially had toxins in the water. And again, this is just a potential, because if you look at the 2018 data towards the right of the graph, we still had that potential signal in the water, but we saw very little toxin production. So it's not really a false positive, but it kind of gives you a sense of, well, the cyanobacteria is there and it could produce toxin, but it doesn't tell you that it is producing toxin. So Dr. Liu took this one step further, and he analyzed reverse transcriptase qPCR. So we're looking now at the RNA, not just the potential to produce toxins, but the gene itself is turned on and it is working. So we're actively producing toxins now. And what you can see here, again, prior to seeing toxins in the water by ELISA, we saw a signal from this RNA. So there was actual genetic indication that toxins were being produced prior to the toxins getting to where we could detect them in the water. So it's not a gradual slope, it's a sudden appearance, and we think it's a great indicator, an early warning system of what is coming. It's not just the potential toxin production, it's the actual toxin production. And even in 2018, where we saw very, very low concentrations of toxins, we still had that genetic sequence present in the water. So we still knew that toxins were being produced, even though the concentrations were minuscule. And here's just a kind of a picture of everything all together. You've got the DNA and the RNA on the bottom graph, and you've got the traditional cyanobacteria, or the traditional phycocyanin and chlorophyll on the top, with the red ELISA microcystin concentration. And you can see all of these DNA and RNA, they give us an earlier warning than even the phycocyanin in the water, even earlier than the satellite signals. So as we continue the work, we're going to further develop that RNA and DNA here in the region. We're going to develop the LC-MSMS toxin analysis here in the region. We're working with our FlowCam on cyanobacterial identification and quantification. We've developed a lake profiler in the region that can send our sondes up and down in the water column so we can get a better idea if the cyanobacteria happen to dive due to rough conditions or no sunlight on top. And then in 2021, our big focus is on urban lakes. We're going to study urban lakes here in Kansas City, as well as Denver and Cincinnati, and kind of see what's so different about the urban lakes and their HAB response. And that is it. If you have any questions, I think we're going to have a discussion at the end.